Good evening, I'm Sashay Evans, and today I'll be presenting on other blood group antigens. Let's go. Did you know that there are over 200 blood antigens? We are used to types A, B, AB, O, and even the rhesus factor. But today, we are going to look at a few rare types. The Lutheran antigen. The Lutheran blood group system was first reported in a patient with lupus who received a series of transfusions and developed a remarkable succession of antibodies. The Lutheran system now comprises of 19 antigens numbered LU1 to LU21, with LU10 and 15 being designated obsolete. The Lutheran blood group antigen has two codominant alleles, LUA and LUB. They are weakly expressed on cord cells because they are poorly developed at birth. Most of the 19 antigens are high incidence, meaning more people are likely to have the LUA and LUB antigens. Lutheran antibodies. The LU anti-A can be triggered without red blood cell stimulation. It can also be triggered by the stimulation of transfusion or pregnancy-related red blood cell exposure. They can either be IgG or IgM. They react at room temperature and are not clinically significant. They also show mixed field reactions. If a woman who has the antigen A on her red blood cell becomes pregnant for a man who has the antigen B on his red blood cells, the antigen and the antibody will react, causing agglutination, which will result in HDN in the fetus. In the case of blood transfusion, a person with the LUA antigen on their red blood cells cannot receive blood from a person who has the LUB antigen on his red blood cells. Bennett-Goodspeed antigen. These are associated with the HLA antigens and are class 1 products of the gene of the major histocompatibility complex. HLA-A, B, and C are present on virtually all nucleated cells. Mature human red blood cells do not have a nucleus and do not generally have easily detectable HLA antigens on their red blood cell surfaces. However, certain HLA antigens are occasionally expressed strongly enough to be detected by conventional blood grouping techniques. These red cells HLA antigens are called BG antigens. BGA represents HLA-B7, BGB represents HLA-B17, and BGC represents HLA-A28. They are not clinically significant, which means the human leukocyte antibodies have very rarely been implicated in acute and delayed hemolytic transfusion reactions. Let us look at the STA antigens. These are high incident antigens found on tissues and in body fluids. The antibodies are not clinically significant and they also cause mixed field agglutination with reagent cells. Another rare blood antigen is called the XG antigen. Only one exists, known as XGA. Inheritance occurs only on the X chromosome, and this antigen is mostly found in women. The antigen is not strong, and so it is not attributed to transfusion reactions. The last antigen we will look at today is the HTLA antigen. The antibodies are very weak and are not clinically significant. These do not cause hemolytic diseases of the newborn or hemolytic transfusion reactions. Let us look at cold antibodies or IgM. Anti-LEA, anti-LEB, anti-I, anti-P1, anti-M and N. Or warm antibodies or IgG are RH antibodies, Kell, Duffy, KID, and SS antibodies. Another way we can identify these antibodies are by their enzyme activity. The KID Rh, Lewis, I, and P antibodies are enhanced by enzyme activity, while FYA, FYB, M, and SS are destroyed by enzymes. Thank you.